eukaryotic transcription. So that was a relatively easy thing to understand. Eukaryotic transcription is much more complex because, as I told you earlier, yesterday, that there are only 800 genes approximately in prokaryotes. In contrast, in eukaryotes we are having 1 lakh genes, and in humans we are having 2800 DNA associated proteins, out of which 1600 are basically transcription factors. So nearly 10% of the gene transcribe transcription factors, and they constitute the largest family of proteins. Can you imagine, out of 2800 proteins associated with DNA molecule, 1600 are transcription factors. So a huge number of transcription factors are involved. In prokaryotes, the transcription is usually polycistronic, which I forgot to mention yesterday. Polycistronic means 2 or more than 2 genes or 2 or more genes are transcribed simultaneously. And prokaryotic genes, prokaryotic transcription, I would say, there is no post-transcriptional modification, and after transcription, the mRNA starts synthesizing proteins. In eukaryotic, it is monocistronic. Only 1 gene is transcribed at one point of time, and that gene is having exons and introns, that is coding part and non-coding part, that need to be removed, and then it has to undergo post-transcriptional modification within the nucleus itself before it is allowed to pass to the cytoplasm. And this complex process takes quite a lot of time, and as I told earlier, that during replication the whole genome is replicated. In contrast, in transcription, part of the DNA, that is genes which are transcribed, suppose in a case when we are in the fed state, by this time you know that after a fed state glycogenesis is stimulated. So the enzymes which will be involved for glycogenesis, which are essentially protein in nature, will be stimulated. Similarly, when we are having a fasting condition, that is glycogenolysis, that is fatty acid breakdown. So all the enzymes related to glycogenolysis will be produced at a much higher speed than what normally it is. And all these mRNAs are having a varying lifespan. So depending on that thing, that lifespan, stability requirement, and need of the cell at a given point of time, there must be a regulation. And prokaryotic genes are not so much of regulated for transcription, but eukaryotic genes are really involved because there are some enhancer sequences, there are some activators, and they play a good amount of regulatory roles, and they are placed in the regulatory region of the RNA. The basic mechanism of prokaryotic transcription and the eukaryotic transcription is same, that is initiation, elongation, and termination. In prokaryotic, what we have seen, that there is only one RNA polymerase which is composed of only 5 subunits. So there are 2 alpha, 1 beta prime and 1 gamma. And when it is associated with sigma factor, then it becomes a hollow enzyme and then its function, and that particular RNAP, that is RNA polymerase, is having all in one function, that is it is having an unwinding capacity, it is having a polymerization, and it is also having excision, that is endonuclease activity also. In contrast, eukaryotic RNA polymerase are not one. There are 3 RNA polymerases, RNA polymerase 1, 2 and 3, and in place of sigma factor, what we have seen, that a number of transcription factors are involved, out of which some are general transcription factors called GTFs. So they come and play into action in place of sigma factors. And in contrast to again that RNAP of prokaryotes, the RNAP1, RNAP2 and RNAP3, they consist of at least more than 12, that is RNAP unit contains 12 subunits. So RNAP1 13 and RNAP3, that is polymerase 3, 17 units. So a huge number of proteins are involved for these 3 enzymes. Now I will tell you in detail. The only difference that is basically DNA remains in a closed structure, in double helical structure, in eukaryotes also, and there is pre-initiation complex, there is promoter binding, and then closed loop is opened by helicase activity, and then transcription starts, the promoter region is cleared off, and then chain elongation takes place, and finally at one point of time termination signal appears. This termination signal, yesterday you have seen, it was basically a CG complex and a polyutil. Here it is different, here it is basically AAU, AAA and CA, which actually recruits cleavage factor to perform endonuclease activity so that the growing or freshly formed or nascent mRNA is released from the DNA, and DNA again winds back to its original position for future transcription. So basically what I said yesterday, so there are 2 strands. One is, this is coding strand, See what we have seen yesterday, there is one transcription start site which was present is +1 basically. This is upstream 5' end. This is upstream as if it is closing to the starting point of the river. So minus, it is designated as minus sign, and that is downstream, is close to the sea where the river will basically fall. In prokaryotes, there is -10 base pair TATA box is located. In eukaryote, it is located about 25 to 30 base pairs ahead in this side. And the CAAT region which you have found yesterday, it was located in 35 position. In eukaryotes, it is located at -70 base pair region, and GC is present in about -90. And then there is a proximal promoter region. There is also distal promoter region, and there is 5' untranslated region. Same thing is there in the 3' untranslated region. These are part of the actual mRNA molecule. Only 3' flanking, 5' flanking region and 3' flanking region are removed. Remember that I will show you in one slide. So basically, the primary transcript is formed, which includes this part also. So transcription starts right here. So basically, location, the first point of contact is at, earlier it was at CAAT, now it is at the GC region, because this will be basically a little bit ahead of this stream. So they will start scanning, and earlier what happened, the sigma factors used to scan, but the eukaryotic polymerase will not be able to do that thing, for which these transcription factors are required. So transcription factor, there are some general transcription factors, and there are specific proteins also which are associated with it. So they will be recruited. Finally, what happens after everything binds, then the transcription starts. So basically, here it is a closed loop. Now there will be opening. There will be opening of this, means this unwinding will occur, and it will form a bubble. Just like yesterday, it will form a bubble, and since this is the template on which this newly formed, that is, transcription will start, suppose, this area. So one nucleotide will be attached to each other. So through phosphodiester bond, after a short stretch of RNA has been formed, then the RNA polymerase 1, just like yesterday, it will move a little ahead to make further synthesis. That means elongation. So after this region, after synthesizing a short part of RNA, the promoter region will be cleared, and all other transcription factors associated with it will also be released. As I will tell you shortly, what are the transcription factors, how they attach. And only see here what happens during this process, this TATA. TATA means the binding protein here is TBP. TATA binding protein, write down. TBP is an universal transcription factor, because TATA box will be there, so TATA binding protein will be there, and TATA binding protein is associated with GTF2D. See, there are general transcription factors. GTF, commonly it is abbreviated as GTF, that is general transcription factors. So GTFs will be there. So this is transcription. TF means transcription factor. So there are at least 6 transcription factors which are common. One is transcription factor 2, A, 2B, 2D, C is not there, 2D, 2E, 2F and 2H. These are the 6 common general transcription factors, and TF2D is basically associated with TBP plus 14 other transcription associated factors. This is last. So this TF2D, this basically comprises this one. So there are 15 elements in Harper book. Yesterday, out of sheer curiosity, I checked the internet and I found it to be 16. There is not this 14 GTF, there are 4A, 4B, and there are 15 is a number of GTF that has also been identified. So anyway, remember this thing. So this TF2D, once it binds to the TATA binding site, this TBP, once it binds, it basically involves TF2D, and there is a sharp bending. There is a sharp bending. This TBP causes bending. That I will show it in a. And once TBP binds, it binds TF2D along with other factors, and sequentially what happens, basically factor D, factor A, factor 2D, B, all these transcription factors they get attached to each other. So transcription factor V, D basically contains this TBP, then the factor A, factor B, basically they stabilize. This role of factor A is to stabilize the bonding between TBP and B, and E is endowed with the RNA polymerase 2. And then H1 is endowed with helicase activity, means this will cause unwinding. This is also having a CTD activity, means carboxy terminal domain of the RNA polymerase will be phosphorylated. Phosphorylation of CTD region that will make the RNA polymerase active. Also, it is having a role on DNA excision that you will read during replication also. Then transcription, there DNA, if there is a false nucleotide which is incorporated, so that nucleotide will be removed. After all these things has been attached, so RNA synthesis, this promoter, that RNA polymerase 2 along with all these factors, DAB, EFH, they will attach to each other and all other factors will be released. Only that factor RNA polymerase 2 that is required for polymerization will remain and it will start producing elongation. So, elongation will occur based on as per the direction of this is template strand, sorry. This is template strand. So, there is growing chain of mRNA, some part has been synthesized. Now, next part will be synthesized as per sequence of the template strand. Now, this will grow. So, at one point of time, you will have this RNA primer RNA, and then the termination signal will appear. And this termination signal basically contains AAU, AAA, and CA. So, this part. So, this AAU, AAA, and CA. This part basically invites cleavage and polyadenylation specific factors. This is called commonly CPSF, and this CA will invite CSF, that is cleavage stimulating factor. So, when now you can understand that cleaving has to take place. So, and in RNA, after the primary transcript has been formed, polyadenylation also takes place. So, cleavage and polyadenylation specific factor, this is also again a transcription factor. So, they will bind. And you see the binding is loose here, AT, that bond is a double bond. So, that will be easy to break also, unlike this bond which is having a 3 hydrogen bond. And CA is cleavage stimulating factor. So, they both will recruit the cleavage factor. They both will basically make cleavage. Cleavage will occur. How this will cleavage will occur? The cleavage will occur because you see this RNA is having this phosphodiester bond within this part. So, one endonuclease must be there. So, it will be endowed with the property of endonuclease. When this terminal part is cut, that is called exonuclease activity. When anything is cut in the middle of any RNA or DNA molecule, that is called an endonuclease. Remember, so restriction endonucleases are there, that we will be reading during recombinant DNA technology, where there are several restriction endonucleases are used. They cut at different points of time. Specifically, exonuclease cuts at the extreme end, it may be 5' end, it may be 3' end, but endonuclease cuts, well, it is in the middle, since it is in the middle, so that needs to be cut, so the cleavage will basically have an endonuclease activity and the nascent or growing RNA chain will be clipped off. So they will come, that will appear as a RNA strand and the RNA polymerase 2 will be released, and these double stranded, these strands of DNA which were unknown will wind back to its original position. So this is in short basically the transcription in eukaryotes. Now you see the slide that it will be a little bit more clear. This what I have said is all these things will be shown in the slide. So yesterday, now you understand the difference between the two, prokaryotic and eukaryotic. This is easy to remember, so that you remember all the points. One is enzyme, that one single enzyme is required, that is DNAP, RNAP, that is DNA dependent RNA polymerase. In contrast, it is having 3 RNA polymerase 1, 2 and 3, and just to remember at least minimal information, that RNA polymerase 1 synthesizes tRNA, mRNA is synthesized by RNA polymerase 2, and tRNA is from RNA polymerase 3. There are other RNAs which are formed, that I will tell. Now here I told you that sigma subunit is there in the prokaryotes, but here it is not so. In the transcription factors, they replace the sigma factor. So TATA binding protein is an universal transcription factor. The start signal there, it was a TATA box basically. It's a TATA box here, it is called a Pribnov box, and the same TATA box is called Goldberg-Hogness box in eukaryotes. So termination, as you learned yesterday, that it was row dependent or row independent. Same thing is there, it can be there, but some kind of lariat structure is there. You see the lariat structure. This is what is called lariat. So that may be there because there are multiple transcription factors. So all these factors will not follow the same thing, and there will be, this is a very brief, in short, a description to have an idea. And there are, you believe, you will start appreciate that there will be a huge amount of regulation. So the regulatory region is present basically located upstream towards the 5' flanking region mainly, which causes enhancer, means which will increase the transcription. There will be silencer, when it will replace basically the transcription. There will be activators which will activate. These can be located downstream also. As I have in the initial picture, I have shown this enhancer may be present on the 5' flanking region or the 3' flanking region, because that also acts as a regulatory part. So regulatory regions are beyond the promoter binding regions. So that basically regulates the entire process. So here it is, a lariat structure is formed. So that is basically AAU, AAA and then CA. You just remember this part, this much. So post-transcriptional modification doesn't occur at all, because it is synthesized and immediately protein synthesis starts, but in eukaryotic it is not so. There are several post-transcriptional modifications like 5-methyl guanosine capping, polyadenylation, there is splicing also, because introns are to be removed, otherwise non-coding regions cannot be included within the protein coding region. And action of antibiotics, I told you yesterday, as a biomedical importance. So while we are treating a patient suffering from some infection due to prokaryotes, we need to get rid of those prokaryotes for which we give the antibiotic. And in cancer also, we want to, no, it is not in prokaryotes, but in eukaryotes, we want to get rid of some cancer. So, we want the cancer cells to die. So, for which anti-cancer drugs are used. Alpha amanitin, for your examination purpose, I will tell you. This is used for research purposes. In a short file, I will explain where alpha amanitin is used. So, the classes of RNA are a huge number, as I can told you out. So, their heterogeneous nuclear RNA means this is a primary transcript which is formed from the actually coding sequence, that part, that region, this transcribing region, the primary transcript which is formed is called the heterogeneous. That is an unmodified RNA molecule. There is small nuclear RNA, there is silencer RNA, there is hnRNA, microRNA, then there is tRNA diverter. There are many things. So, you see classes of eukaryote by and large, you see the mRNA, which is having, we are having 1 lakh genes, so almost nearly more than 10 to the power 5 means 1 lakh mRNA position. There are the stability, again it may be very unstable to very stable. Those who are unstable, they don't require frequent synthesis, they require frequent synthesis, but those which are stable do not require so. So, if from the chart you can understand the different types of eukaryotic DNA, their abundance. This rRNA, which is present in the most abundant, 80%, and it is a very stable form. And there is INC, LNC. This is, remember, this is LNC means large non-coding RNA. And snRNA, small nuclear RNA, microRNA, or silencing RNAs. So, they are having different roles. What are the mobile? snRNA. So, snRNA is having some of the small snRNA, as an example I have shown, that usually the nucleotide are present in snRNA is about 150, but the range is quite wide, 90 to 300. And you can see some of the ranges, it is 95, 45S, and you can see 216, also 280. So, their presence is either in the nucleolus, nucleoplasm, or it can be nucleus, or it can be both nucleus and cytoplasm. So, I told you this eukaryotic RNA polymerase contains 5 units, 2 alpha, 1 beta, beta prime, and 1 omega. In contrast, and they produce 3 RNAs only, rRNA, mRNA, tRNA. In contrast, you see the RNA polymerase 1, as I told during this, before showing the slide, that RNA polymerase 2, which is concerned with the mRNA synthesis, is having 12 subunits. This is only 5. This is 12 subunits minimum. RNA polymerase 1, 13, and RNA polymerase 3 is 17. That means about 42 proteins are involved in RNA polymerase, in contrast to only 5 of the prokaryotes. What is that? So, you see the eukaryotes means starting from yeast to human being. So, this is a wide range of eukaryotes. So, this is what I have shown you is from mammalian nuclear DNA dependent RNA polymerase. So, what they produce? This RNAP1 rRNA, RNAP2 mRNA, this you at least this you must remember, this RNAP2 rRNA, tRNA. The amanitin is basically a peptide which is a toxic substances. It is used for research purposes. Now, why it is important? Suppose you see the sensitivity, RNAP2 this is high sensitive, RNAP1 it is totally insensitive, and this is intermediate sensitive. RNA polymerase 3. Now I want to do a research activity. I want to see whether rRNA is formed from RNA polymerase 1 or not. Now we have added one test tube RNA with RNA polymerase 1 only, and in another test tube the same quantity with alpha amanitin. Now those test tubes which will contain alpha amanitin, since this is totally insensitive, there will be basically no change. Thus, if I try to synthesize mRNA or log non-coding RNA or mRNA, siRNA, and if we add this thing, since this is high sensitivity, that means this will decrease. So in one test tube we will have these functions of this mRNA form, in other test tube we will not get it. Since this is having intermediate sensitivity, at least some part of formation, some formation of tRNAs will be there, but not with the control tube. Control tube is not having this thing, alpha amanitin. So this is basically done for research purpose. So examiners are fond of asking this type of questions. So you remember this as an MCQ or just for VIVA. Now there are other RNAs like RNA polymerase 2,1 also produces 28S, 18S and 5.8S. RNA polymerase 3 also produces 5S. Human ribosome is basically a 8Ts ribosome, but this is composed of 60S and 40S. Similarly, bacteria 70S, it is 50S and 30S. Why? What does this S signify? Try to remember, this is expressed in Sefberg units. What is Sefberg unit? Basically, as you understand that while you centrifuge, you remember the subcellular fractionation of organelle separation. Have you read it? How many of you are aware of it? That what is subcellular fractionation? How it is done? Can anybody tell? Can anybody tell how subcellular fractionation is done? I mean, how I will get mitochondria? I want to have a preparation, not the whole cell, but only the mitochondria, or only the ribosome, or only lysosome, peroxisome, endoplasmic reticulum. I can get it. To do an experiment, how will you get it? That means there is a question, none of you have read it. So, by doing a centrifugation, different cell centrifugation at different speeds. So, if you, centrifuge it at more than 1 lakh G, you will get the cytosol, means all the subcellular organelles will be removed, you will get the cytosol. As you increase the speed, those which are having a larger size will get deposited, sedimented at a lower speed. So this is basically based on the sedimentation rate, and usually is an ultra centrifuge. So a high speed centrifuge is used, and based on the sedimentation rate, these subunits are classified. As I told you yesterday also, only portion of DNA is transcribed, not the whole thing, and that depends on the cellular need at a given point of time. So because mRNA is stable for a variable period of time, and if there is any problem in the post-transcriptional modification or splicing, there will be disease. What देखो तो एरा less होगा. What देखो. So complex eukaryotic protein, I told you there are 2,800 proteins in the human genome, and 1,600 of these are associated with transcription factor, that is 10% of the genes, and that constitutes the largest family. See, all polymerases must bind to a specific site to function. See, RNA prokaryotic is endowed with all properties, but DNA is not so, so they require growth factors. This transcription, gel transcription factor, as I told you, to catalyze specific transcriptions. The role of prokaryotic RNA polymerase is assumed by the transcription factor. The sigma factor, which was present in the prokaryote, has been taken over by the transcription factors. So 12 subunits I told you of RNA polymerase 2 requires 6 GTFs. So these 6 GTFs, as I told you, A, B, D, E, F, H, and that contains about 33 additional polypeptides. So TATA binding protein, that is called TBP, it is a universal transcription factor, and I also explained that TF2D complex is consisting of about 15 subunits. One is TBP and 14 TAFs, that is transcription associated factors, and I checked it yesterday. So it was total number has increased. So in the next edition of the book, while you will be in a final year course, at least for examination purposes, check what is the latest figure in the book and also in the internet. So because these are the, high level questions that are usually set during the examination. So only remember one thing, this TF2D is the only gel transcription factor that is capable of specific high affinity binding to promoter region of the DNA. So transcription control elements, the number, only TATA box, CAAT box, GC box I have shown. There are other boxes, there are other regions. It is not that these 3 regions which I have shown here is the only controlling agent. So a number of factors are there which are affected. You can see the several members of this family. I have indicated only a few. Regina, mobile dekho na. Basal component, there are transcription factors. So these 6 factors, this RNA polymerase 2, TATA binding protein, A, B, D, E, F, H, these you must remember. I have repeatedly told this at least 6 to 7 times. Now, co-regulators are this TF2D, is basically TBP plus TAF, 14 TFs. There are certain genes, mediators, and activators are SP1 means special protein 1. There is ATF, CTF, and there are associated proteins also, AP1. But যে ছবিটা আগে দেখাচ্ছিলাম at the beginning, you see the region, this is the start region. But this part, this 5' untranslated, remember, means this part is not translated. This is 3' untranslated region. That means this is not translated during protein translation, that means protein synthesis. But these are included during post-translational processing, because 5' capping is done and poly A tail is done after that. So that part which is there, 5' untranslated region, is also a part of the primary transcript, but this is not the protein coding region. Protein coding region is this one, which is composed of exon and intron. Introns are removed and exons are spliced together. So translation will occur with the protein coding region, not with the untranslated region. Name is untranslated, means this part is not translated. So don't get confused with the 5' flanking region or 3' flanking region with the 5' UTR and 3' UTR. 5' UTR is totally different than 5' flanking region. 5' flanking region is mainly concerned with the regulatory elements, regulatory activities of mRNA or tRNA, these transcription factors, basically transcription. So 5' and 3' flanking regions are basically the regulatory regions. Don't get confused with the 5' untranslated region. and 3' UTR region with the flanking regions. So this is basically 3 steps as I told: initiation, elongation and termination. This is what happens. This I told you, again just a revision, recapitulation. So 25 base per upstream TATA box, 70 base per upstream, that is CAAT box or proximal promoter element, there is distal promoter element also there. Beyond 90 base per upstream, there is GC region, then formation of pre-initiation complex, then initiation of RNA synthesis, then the area will be cleared of the promoter clearance will be there. So that is basically initiation. So initiation, now you see that part. So that includes basically transcription unit is having that promoter region, that is TATA box, CAAT box, GC box and other elements. TATA box is located 25 to 35 base pairs upstream, GC box is minus 90, minus 75 and GC box is 90, definitely CAAT box is 75 base pair upstream, and they help in the regulation of RNA polymerase 2. So key features, these 3 boxes I have already explained. Transcription start site will be there, RNA polymerase 2 will be required, but they will not, the RNA polymerase 2 will not be able to function properly because of without joint transcription factor, and one part is having that catalytic activity, that is H activity. H basically having 3 functions. One is helicase activity that unwinds, and then it is having a CTD phosphorylation which will activate the RNA polymerase, and third one is nucleotide excision repair. Now you see that GC region, I told you GC region usually is present beyond this region. Now at times it can be present here also. So from this is a thymidine kinase gene, so TATA box is there, but here GC box is there, GC box is also here. So everything is not by default, it has also come to an end. And just if I told you that this TATA binding protein that causes bending, say number of activators are there which are located upstream, they are bound to each other. Suppose this is gene A, this is gene B, and they bind to each other, and this is distal control element, and this is the region of DNA bending protein, that is TATA binding protein. And once this happens, I told you that sequence DAB, EFH, that you see is this TAF is basically having binding, tightly binds with the RNA polymerase 2, and TFIIH is having highly phosphorylation, and this you understand this part. So now you see these structures are getting attached to each other. So initially before the helix opens, that was the structure which is present, and RNA polymerase 2 binds, and after this, by bending, all the factors, all the transcription factors get attached to RNA polymerase 2. So bending up to unwinding, and then initiation of RNA synthesis, promoter clearance. This is basically this initiation part.